Katie, thank you for accepting being among us in the symposium. I will give the audience a short presentation of you, and then we can start the talk. So Professor Cullen received a PhD in 1991 from the Committee on Neurobiology. Her advisor was Robert McRae. Dr. Cullen was a fellow at the Montreal Neurological Institute, where she worked in the Department of Neurology and Neurosurgery. In 1994, Dr. Cullen became an assistant professor in the Department of Physiology at McGill University, with appointments in biomedical engineering, neuroscience, and laryngology. In 2002, Cullen was appointed a William Dawson Chair in recognition of her work in systems neuroscience and neural engineering, and served as director of McGill Aerospace Medical Research Unit, comprising 4 faculty and their research labs. In 2016, Dr. Cullen moved to Johns Hopkins University, where she is now a professor in biomedical engineering and holds joint appointment in the departments of neuroscience and in otolaryngology, head and neck surgery. Kathleen, general areas of interest is in systems, computational neuroscience, with an emphasis on translational approaches to restoring sensory function. Her research expertise lies in understanding the coding strategies that the brain uses to create neural representations of self-motion. She studied how the brain combines information from the vestibular sensors with extravestibular cues, visual, touch, and motors, to estimate self-motion to ensure accurate motor control and postural stability. One of Kathy's main findings has been the identification of a distinct and surprisingly a small cluster of cells deep within the brain that react within milliseconds to readjust for movement when something unexpected happens. The experiment that leads to the discovery devised by Kathy and one of her former PhD students, Jess Brooke, involved putting macaque monkeys inside a device similar to a flight simulator. And this finding actually both overturned current theories about how we learn to maintain our balance as we move through the world, and also has significant implication for understanding the neural basis of motion sickness. Actually, this study was published in Current Biology and was widely covered by the media. Okay, Kathleen, we are looking forward to have your nice presentation, and thank you again for accepting our invitation to be part of this symposium.
Yeah, well, thank you very much for the invitation to participate, and I look forward to sharing my science with you and to discussing things in the panel after the presentation. Okay, so I'm going to start with a few general comments that I'm sure all of you are aware of, but just to kind of set the stage. So in everyday life, our sensory systems are activated simultaneously by both sensory stimuli that result from changes in the external world, but also as a result of sensory inputs that are a result of our own actions. So those sensory inputs that are a result of external events are called Afferents, and sensory inputs that are a result of our own behavior are called Afferents. And the ability to distinguish these two types of sensory inputs is essential both for our perceptual stability, which I think people have thought about for quite a long time, but also for accurate motor control. And I'm going to make a case for that in the context of the vestibular system, which is, as was already noted in the introduction, a focus of our lab's work, where we've really tried to understand how vestibular information is coded during active behaviors, and then specifically look at the mechanisms that underlie the distinction of vestibular Afferents from reafferents during natural self-motion. So by way of introduction, because I appreciate everybody is not completely familiar with the vestibular system, from an engineering point of view, it's really a beautiful system. You have effectively a three-dimensional gyro on each side of your head and a three-dimensional accelerometer. So three-dimensional gyro is the semicircular canals, which sense head rotation in three axes, Cartesian coordinate system, therefore can sense all rotations, in all directions by the ratio of activation of the three canals. And in addition, we have two otolith sensors, which sense linear head motion, including gravity. And they make up basically the neural epithelium is organized such that it encodes three-dimensional linear motion. So from an engineering point of view, it's really quite a beautiful system. And individual afferents that travel in the eighth nerve, which is shared with the auditory system, innervate only one end organ, one of the five. So you can get really these beautifully clean signals that are representations of either angular head rotation or linear head acceleration in a particular dimension. So when people think about the vestibular system, it's generally synonymous with the vestibular ocular reflex, which is indeed one of its primary functions. So if you didn't have a vestibular system, life would be very uncomfortable as you do your everyday activities, and it's something we really take for granted. It's working in the background for us and keeping our world stable and giving us our perception of how we're moving through space. So the next slide I have shows an example movie that my postdoc in Montreal took with one of these old GoPros that didn't have gaze stabilization in it as he's walking down St. Catherine Street in Montreal. And hopefully this will work. Oops. This might, okay, I think it'll work. So he's just walking. Right, so it's not a particularly rigorous thing that Jerome is doing in this video. Is just walking normally, but you can see there's all this bouncing, right, and blurring of the visual world, which we normally don't see when we walk down the street. And that's because as we walk down the street, for every head movement our eyes generate equal and opposite eye movement so that the visual world stays stable on the retina. So that's one function of the vestibular system. I'm not going to focus so much on that in my talk. I'm going to focus more on a couple other functions. So one is the maintenance of head and body posture. So if you don't have a vestibular system, you're going to be quite compromised in doing any sort of activity that requires good balance because the vestibular system provides very fast, dynamic input, much faster than the visual system can about how we're moving through space. And it's also an important input to our higher level areas in our brain to help us compute our sense of self-motion and spatial orientation. Okay, so my talk is roughly organized into two main questions because I always like to think in terms of questions that are being answered. And the first question is, how does the vestibular system encode natural self-motion? And this is really in the context of maybe a more traditional maybe engineering style approach, computational approach to understanding neural coding. And I trained initially as an electrical engineer in addition to a neuroscientist, so I like to use these types of approaches to really quantify neuronal coding. To me, it's a very rewarding endeavor. But I'll also show you that there's more to it than the sort of traditional approach in the second part of the talk where I talk about the encoding during active behaviors. Okay. So one of the things about the vestibular system is because the head movements are quite easy to characterize, and we have this nice sort of Cartesian coordinate system for head motion, both in terms of rotation and translations in the ear, we can actually use linear systems approaches to make good progress on understanding how information is encoded and processed as we move stage to stage to stage in the system. So in this approach, people use sine waves of different frequencies. The input would be, say, head velocity. It could be rotation or translation. The output could be a neuron that you're recording in the pathway to understand how its responses are dynamically, as a function of time related to the input and the frequency dependency of that relationship. And when you characterize that, you basically are estimating the transfer function between the input and the output. And this has been a very dominant approach in vestibular and ocular motor work. So in our experiments, what we do is we go in, and this is actually done a lot of this in rhesus monkey. We also do some work in a mouse model, and record from single afferents using fine glass micropipettes that are actually single fibers within the eighth nerve, the shared nerve with the auditory and vestibular system. If you go into this nerve, what you'll find is there are two classes of afferents when the animal's not moving. One class has very regular clockwork-like firing so that the inner spike interval is very fixed. So on average, afferents fire about 100 spikes per second. So the regular afferents would pretty much fire like clockwork every 10 milliseconds. And then in addition, you have the second class of afferents called irregular afferents. Irregular afferents have the same average resting discharge, but their resting rate variability is much higher. So you can see their inner spike interval, if you plotted a histogram, would be much broader. So what we can do is rotate an animal, for example, if we're recording from afferents that are innervating the horizontal semicircular canal, as we rotate the animal back and forth in the horizontal plane. So we can have horizontal head rotation here in terms of velocity, and then we can look and see what the afferents are actually encoding. One thing I'd note is that when we started this work, we knew that these two types of afferents actually innervate separate classes of receptor cells in the neural epithelium within the sensory organs. So these are hair cells that are a bit like what you would see in the auditory system in the cochlea. The type 2 hair cells actually look a lot like cochlear hair cells, but the type 1 hair cells are actually unusual. Here you have an afferent projection that goes all the way up around the cell body of the neuron, and in contrast to this more conventional bouton ending that you have at the base of the type 2s. So if you look at this cell body of the receptor cell and its afferent innervation, what it really looks like is it's built for speed, right? This afferent is not taking any chances in really getting very quick and reliable information from that cell body. And what's interesting is that this second channel, these type 1 hair cells, evolved later than the type 2 hair cells. So if you look at animals like fish and amphibians that still live in water and lived in a more viscous environment, they have type 2 hair cells. But if you look at animals that have moved on, higher vertebrate classes that have moved on from sea to land, you see basically the evolution of this type 1 hair cell. So that's kind of a clue about what the two channels might be doing. So again, these type 1 hair cells are primarily innervated by irregular afferents and the type 2 by regular afferents. So it looks like this irregular channel is more recently evolved when animals move from sea to land. So if we actually play in this sort of rotational stimuli and move our animals on our rotational device, and we look at regular versus irregular afferents, what you can see is for the same amplitude head velocity, as you increase frequency, you get a big difference in the firing rate. So you get an increase in firing rate with increasing frequency, and this increase modulation is even bigger for the irregulars than for the regular afferents. The other thing to note is how beautifully these afferents actually encode the stimulus. Okay. So what we can do then is plot as a function of frequency the gain of the response modulation gain, and you can see that across this range, which goes up to 16 hertz in this example, the regular afferents are always less sensitive than the irregular afferents. So this sort of raises the question. Remember that this regular channel is the more ancient and the irregular is more recent. Why do we hang on to these regular afferents? What's their utility? Why do we still have them? If these other afferents that are more recently evolved are more sensitive across the frequency range. So to try to understand this, the first thing we really wanted to do was understand, well, what do these neurons need to encode, right? And I realized that this is actually back in, I think, 2010, sort of shockingly, people didn't really know what the natural statistics of motion were during active behaviors or just behaviors you would experience in your everyday life. So this is work done by my student Mohsen Jamali, who's currently a postdoc at Harvard, and my other postdoc, Jerome Carriot. And what they did is we basically built little IMU sensors with a platform from ST Microelectronics. We put a cell phone battery in, a little data card. It was a kind of clunky circuit that we put on people's heads, and we had them running around the city of Montreal on buses, on the metro, playing soccer, doing all kinds of different things, and looked at how they actually really move. And what we were surprised to see is what high intensities people can generate in terms of self-motion during their everyday activities. So movements reach intensities up to 8G in terms of linear motion, 15,000 degrees per second in terms of rotational motion. Movements are highly non-Gaussian, so you have a lot, you can see that down here. Here's a distribution of yaw rotations, horizontal rotations with very peaky in the middle and it has long tails. We find there's significant power up to 20 hertz. And for those of you who know anything about statistics of natural sensory stimuli, vestibular natural input does not follow a power law. So if you plot on a log-log scale power versus frequency, you don't get a nice line. It's actually flatter at low frequencies and then falls off much more quickly at higher frequencies. So there's this sort of two piecewise linear fit you need to do rather than a single power law. So we also then went and said, well, we're not doing our single unit recordings in people, obviously. What we're doing is these experiments are being done in monkeys and mice. So we went back and did a similar parallel study to see how animals move during naturalistic behavior. So we had a couple of monkeys in a big room with lots of things to climb and food and very enriched environment and saw how they moved. We did something comparable with our mice. And the good news is actually monkeys are a wonderful model for human behavior in terms of natural self-motion. So we have a paper in J. Physio London demonstrating that. Mice are actually pretty similar, surprisingly similar. There's one small caveat on that, but it made a good point for the utility of these animal models. So what did we learn when we did that? So I already mentioned that the range over which we move is actually quite big. But if you look at, and this is a good, I think, good lesson. I was sort of saying initially that it's really the use of linear systems analysis has been so prevalent in our field, and it's something that's been very useful for making progress. But we've also learned in order to do that, we really need to restrict our stimuli so we don't drive neurons into their nonlinear range. So if we do that, we're actually stuck in this tiny little range here when we compare that to what you actually see during naturalistic movement. And I think that's important because it's a case in which the tool limits the way that people think about the science. So it's not to say that there's nothing to be gained by the linear systems approach, but if you actually want to understand what a system is designed to do, you might want to understand really what the natural statistics of movement are, or whatever sensory input you're looking at, and then explore that space as well. Okay, so we went back and said, okay, let's look at more naturalistic encoding and take advantage of stimuli that actually we recorded during our other experiments, or like monkeys running around naturally. We can take little epics of that and play it into our hexapod or our different motion platforms and see how neurons record to these more dynamic stimuli. And what I'll just note actually is we can make a prediction, right? So if you look at the variability of the resting discharge of the afferents, and I mentioned if you took an interspike interval and plotted the interspike interval histogram for regular versus irregular, you'd see this very sharp peak for, at say, 10 milliseconds for a regular afferent because it's very clock-like, and a more broad distribution for irregular afferents. We can take the power spectrum of that, and again, this is in the absence of stimulation. This is what these afferents do when the animal's at rest, and then it modulates around this resting discharge. So if you look at the power spectrum, so you're going to say for regular afferent have a peak at 100 hertz and then the subsequent harmonics, 200 and 300 hertz, which you'd expect because it's periodic. And this gray box represents the frequency range over which encoding needs to be done. That's the frequency range of naturalistic head motion up to about 20-25 hertz. So here there's actually very little power at rest for the regular afferent, but in contrast, if you look at the irregular afferent at rest, there's a lot more power there. So what that suggests is you actually need more signal, right, to drive the neuron out of this sort of noise that's seen at the low frequency range over which encoding needs to be done. So the prediction is that the lower power in the regular afferents firing in that gray band, right, which is where the stimuli actually are occurring, predicts less noise and better information coding in this physiologically relevant range. So we went and tested that prediction by, instead of playing in sine waves, playing in naturalistic head movements. And what we can do is then record, this is the top trace is, the gray represents the head motion. The middle trace is the spike density. The bottom trace is the spike train. To get the spike density, we just convolve a Gaussian filter with the spike train to get the spike density curve. But the other thing we can do is that we can actually compute the optimal linear filter that we convolve with the spike train to reproduce the stimulus. And that's what's shown here in the blue dashed line for a regular afferent. And what you can see right away is that these neurons are kind of addicting to record from, right, because they do such a beautiful job of predicting the stimulus that you used. So here you have very good ability based on one single afferent fiber to predict what the stimulus was in the case of the afferent. We can go through that same exercise for an irregular afferent, and what you can see below is that the irregular afferent's coding is actually much worse, right? Using this stimulus reconstruction where we estimate the optimal linear filter that we convolve with the spike train, we can't do a very good job of reproducing the stimulus. So based on the fact, and what you can do is quantify the ability to reconstruct the stimulus and compute something called a coding fraction. And what this suggests is that regular afferents have better coding fractions, right, than irregulars. So this, based on the math, we predicted that the regular afferent should also carry more information in their spike trains. And that's what's shown here. And this is the physiologically relevant range, again, from 0 to about 20 hertz. And what you can see here is that the regular afferents are actually better encoding far more information, right, over that entire frequency range until the very end where they start to cross over with the irregulars. And that's where the irregular gain is getting bigger and bigger. And that again contrasts with this traditional approach that people have been using in the field up until we presented this work, where you basically see that the gains of the irregulars are higher than those of the irregulars. So an easy way to think about this is that the irregulars have higher gains, but they're also much noisier. And in terms of signal noise, the irregulars are actually problematic, and the regulars are better in terms of their signal noise over the relevant frequency range. The other thing we could look at, and this is the end of the part I'll talk about as far as question 1, is precise spike timing. So the idea is that if we use the traditional either linear systems approaches or the approach I just went through, we're thinking more about rate coding than precise spike timing. But if you look at, say, a little snippet of naturalistic movement and play it with our motion platform and record an irregular versus a regular afferent, what you can see right away is there's a big difference in the sort of patterning of the action potentials for the irregular versus the regular afferent. And remember, if we looked in terms of information coding, even though you don't see this patterning in the regular afferent's discharge, its information coding would actually be better. Right? Its gain is low, but its information coding would be better. So this led us to look into different ways to, I guess, investigate whether or not there was a precise spike timing code, meaning that the actual precision in which each spike was placed matters. And what we did is we used a couple classifiers that are in the literature. There's the Van Rossum classifier, which is quite popular, for example, which I think is what this one shows. There are a couple others. And the idea is what you do is you take a number of different naturalistic stimuli, you play them over and over and over again, kind of like you do here. Then we could do this for another stimuli and another stimuli. And you try to determine how well you can, based on the spike train, identify the actual, so what you do, say, we play this stimulus, and then we take this top response as the template. So rather than reconstructing on the stimulus itself, what you're doing is taking the spike trains and seeing how similar they are across different stimulus epochs. And what you do is then compute the performance of the ability to choose properly based on the spike timing precision. And these metrics are basically set so that you are jittering spikes and removing spikes in order to classify properly. So it's a cost metric. And what you can see again here is there's this nice banding for the irregular that's not there for the regular. So you could predict probably that you're going to have more precision in the actual timing of the spikes for the irregular. And that's what we see. And if you're interested in that approach, you can look at Mohsen Jamali's papers. This was work from his PhD thesis. And ultimately what we see is close to perfect performance for the irregular afferents at a time scale of about 5 milliseconds, which is well outside the range of any feature of the stimulus that was played. In contrast, if we look for the regular afferents, their spike timing precision is much longer, 6 times longer, and also the ability to classify is nowhere near as good as it was for the irregulars. So what we see is that there are 2 distinct information channels in early vestibular processing, and here I show for canal processing, we did the same thing for otolith processing. We have regular afferents that use a rate code in terms of mutual information, and then we have irregular afferents that use a temporal code in terms of precise spike timing. And again, what's a key signature of this spike timing precision is that the temporal precision is something that's outside the range, right? Finer than anything that's actually stimulating the neuron, which is the case for this. And then when we went in and looked at the central neurons to see if in fact we could see evidence for either rate coding or temporal coding in the central neurons, we focused so far on the neurons I'll focus on for the second part of the talk, these vestibular-only neurons that contribute to vestibular spinal pathways and higher-level pathways. These neurons actually don't do very good rate coding. They're actually encoding less information than the irregular afferents, which was sort of surprising to us. But if you look in terms of their temporal coding, they show actually excellent spike timing precision, and with the same temporal precision on the order of 5 milliseconds that the irregular afferents did. So that suggests that this temporal code is transmitted to central pathways. So if we think about this in terms of the function of the VOR in that slide I initially showed you, based on what we know about the connectivity of regular and irregular afferents, what we think is the regular afferents, which are this low gain, high information pathway and use a rate code, are involved in central vestibular ocular reflex pathways. That's the pathway that helps you encode stable gaze when you're walking down the street. Whereas this irregular pathway, which is high gain and low information, but it has a very precise spike timing code, is involved in vestibular spinal and ascending thalamical cortical pathways. And I'll just note, and I can talk about this during the panel, that this work actually is, in terms of translation, very important because it informs the design of a more biomimetic vestibular prosthetic. So there are currently ongoing trials at Hopkins with a vestibular prosthetic, and I've been collaborating with people, even when I was at McGill, regarding this work. What we're interested in doing is looking at how, if we have the same prosthetic implanted in monkeys, single neurons are responding and how are they encoding it, to understand how to make the next generation of prosthetic better. And one of the kind of exciting things is all this information that we now know about how afferents encode information inform the mapping function of this prosthetic, of how do we turn, say, gyro motion, which is currently what this prosthetic does. It encodes angular head rotation and activates the 3 semicircular canals by mapping a 3-dimensional gyro into the canal planes. So the question is, how do you map motion that's recorded with that 3-dimensional gyro that's replacing the end organ in the patient into a pulse rate, right? And we can use what we know about the vestibular system based on our basic science to inform that mapping function. So what's kind of exciting is the patients who currently are implanted with this prosthetic have a mapping function based on our basic science embedded in their prosthetic. Okay, so what I want to talk about now, which is probably the work that my group is the best known for, is how does the vestibular system encode self-motion during active behaviors? And this is very, a long-standing question in the field, right? So it's been known back since the time of Helmholtz, who I think made many important observations, that if you tap on the side of your eyelid, you get a little shift of the world. But you never see the world shift if you make a saccade from the left to the right, right? So you have perfect stability in that case. So in the first instance, tapping on the side of your eyelid is a sort of artificial thing. But in the second case, your brain has a copy of the ocular motor command, the motor command that was made to move your eye, and obviously using that to keep things in register. what people probably have thought a little bit less about is that this ability to make the distinction between active and passive motion is also critical in terms of accurate motor control. So in terms of the vestibular system, the same central neurons that get afferent input, so the afferent input from the afferents I just described, the neurons that get that direct input send their axons directly to motor neurons, right? So for example, the vestibular ocular reflex is a 3-neuron reflex, afferent central neuron, extraocular motor neuron. So it's basically 3 neurons. And then it's the same for the vestibular spinal reflexes in their most direct form. You have afferent central neuron projecting directly to the spinal cord. So if you think about vestibular spinal reflexes, for example, they're really useful if you want to stabilize yourself relative to space. But if you actually want to move through space, they're counterproductive. So this is an example sort of based on life in Montreal. This would be somebody walking down the hill from where my lab used to be, about to slip or slipping on ice, hopefully not going to fall because their vestibular reflexes are robust. And this would be an unexpected vestibular input, right, that's helping them, very quickly know, the brain knows that they're moving unexpectedly, and hopefully they'll mount a proper response to stay upright. This is someone who actually has practiced, a jump probably thousands of times, and is maybe experiencing very similar vestibular inflow, right? They look like they're maybe moving very similarly through the world, but you would guess that you would not want her to be mounting a vestibular spinal, robust vestibular spinal compensatory reflex, because that would actually break her jump and make her stop moving. So even though we just have a very short pathway mediating these compensatory spinal reflexes, in one case we want a robust reflex that hold us still and maintain posture. In the other case, we're actually trying to move through the world. So that is a counterproductive reflex. So where do we make this distinction so we can gate out, for example, vestibular spinal reflexes when we want to move through the world? We don't have a lot of space. Like I said, you have afferents, central neurons, and then projecting down to the spinal cord. So the first thing we can do is start and look at the afferents, right? Again, the reason we would do this is because there is a vestibular efferent system that we know projects back out to, projects from the brain back out to the periphery. And there's the long-standing, prevailing hypothesis has been that this system is there to prevent overstimulation of the sensors when you are actively moving. So we decided to look at this and actually expected to see a positive result, but that's not what we saw. So this just shows the way we organize our experiments. So this is an animal making active head movements and to look back and forth at target. So he's actively moving his head through space. We can take that same head motion and play it into our motion device so we can move the animal the same way through space. So his head again is moving in the same exact way through space. So the stimulus of the vestibular system is identical. And what you can see here is afferents again are really beautiful at encoding head motion. So you can see this nice representation of the head motion. This would be a regular afferent. And here you can see that the discharge of the response, or the response of this neuron, is pretty much identical. And that blue trace that's superimposed on the gray firing rate of the neuron represents a model based on the neuron sensitivity to passive motion. So a passive model represents the motion in both conditions. Right? And the model is basically going to be a resting discharge, which again is about 100 spikes per second, some head velocity term, and then a little bit of a lead, because in addition to having modulation that increases as a function of frequency for gain, we actually have a lead that increases with frequency. Okay, so what you can see here is the sensitivity to passive rotation versus active rotation, and then the change in bias for that equation I just showed you in the two conditions. And there's no change in either the sensitivity or in the bias for active movements. And the idea had been based on some work in model organisms like toadfish and xenopus, larval xenopus, that we would expect to see an increase in the bias during active movement, so you'd be less likely to cut off, and a decrease in the sensitivity. And we don't see either of those effects. In more recent work, you could say, well, natural head movements is one behavior, but how about other natural movements? So we've more recently been looking at locomotion. We've been using deep lab cut, which I'm sure many of you are familiar with, which is a markerless tracking algorithm that's based on neural nets. After training, we can actually do a really good job of markerless identification of different joints to keep track of how the animal's actually moving. And what we can see here is passive movements versus walking and running for our monkeys. And the short answer is that just like we saw for active head movements, vestibular afferents do not differentially encode active and passive movements. So they really are just acting like MEMS sensors. They're encoding how the head is moving through space regardless of the context. So they're context independent. So the good news is that everything I told you in the first part of the talk is still true, that we have two coding channels, and that's where we have regulars which use a rate code and irregulars that use a spike timing code, etc. And that this is true regardless of whether head motion is passively applied or actively applied. And again, a difference between what I just showed you and what I showed you in the first part of the talk is in the first part of the talk we recorded naturalistic movement, but then we played it in with our sensor, with our turntable, right? So it was passively applied, though it had features of natural movement. And in this part of the talk, the animals are actually generating the movement. Okay, so again, we said, okay, we want to have this differential representation of active versus passive movement, but we're not seeing it at the level of the afferent. So let's now go one stage further into processing and look at the central neurons. So these are the neurons that get direct afferent input, and we know the afferents are encoding a vertical, right, a true representation of how the head is moving through space. What's happening at this level of this first central neuron? So this is what I've already showed you. We have passive movements, right, and we have this replay of active movements. And these neurons, which again are called vestibular-only neurons, in part because they don't have eye movement sensitivity, are neurons that are involved in the vestibular spinal pathways and also send information up to the ventral posterior lateral thalamus, and which then goes up to places like PIVC and other multimodal areas in cortex that integrate vestibular information with other multimodal inputs. Okay, so again, these neurons are nicely sensitive to passive applied motion. They look a lot like afferents, but we see something really fundamentally different in this next stage of processing when the animals generate their own active head motion. So what you can see right away, the gray represents the neurons firing. The head motions are identical as they were in the passive condition. The dotted line represents how much that neuron should have fired if it had the same sensitivity it did during the passive condition. And what you can see is this profound suppression. This is almost 100% suppression for this particular central neuron. And then we could do another experiment. I actually love this figure where we have the monkey rotating passively on his turntable while he's actively generating movements. So he's generating active movements while we passively rotate him. We're passively applying a sine wave that's shown in this dashed gray line. His active head and space motion is this black line here, and then total head and space motion is the black line up here. And it's maybe easier to see on the right side. So total head is black, the gray dashed line is the passive component, and the difference between those is his active component, which is the black line here. So the question is, do the neurons encode total head and space motion, as an afferent would, or are they just selectively encoding the unexpected passive component that we're applying? And that's what you see right below, is that the neurons are tracing out only the sinusoidal component of the motion, which is what we are externally applying, and they're totally ignoring the self-generated component of the movement. So this is a pretty profound computation, right? Because again, if you thought about the afferents, they'd be following that dashed blue line. They'd be encoding total head and space motion, just like a, like a IMU sensor, or like a gyro sensor. So if we look at the circuit again, what we see is there's a green line coming in here, which means the afferents are carrying all head motion information during active movements. So somehow, since these central neurons are getting all head motion input, what we need to do is cancel out specifically the component of head motion that the monkey generated itself. We need to find a way to cancel the reafferent vestibular input. So, and I should note that what we see for the rotational examples I've been going through is not specific to horizontal head rotations. We see it for linear motion. We see it on otolith afferents are likewise suppressed. We see it also across different species. We see it in mice. So it's a very general principle, this reafferent suppression at the level of these VO neurons. So the question is, how is that suppression occurring? We have whole body rotation is activating the vestibular system, but during active self motion, we have these other inputs. We have proprioception, motor command, and we have the knowledge of self motion. So we actually went and looked through these three other possibilities one by one to try to dissociate their effect. So what I can tell you is that none of those three additional inputs during active self motion is sufficient to account for suppression. So to look at the activation of proprioceptors, because if you move your head on your body, you stretch your neck. What we did is we held the animal's head fixed in space and rotated its body underneath. So that's what you can see there. And in monkey, these neurons were never activated by this stimulus. So it's not just that we're activating neck proprioceptors when the monkey moves his head on his body. We also looked at the generation of motor command. So the idea is that the monkey's making active head movements, and when he doesn't expect it, we put an electromagnetic clutch on his head, so his head is then stable in space, but we can measure the neck torque against the restraint. We can also see he's trying to look at the targets because he's making eye movements. And even though there's torque, which shows that the monkey's generating a neck motor command, we never saw any sort of inhibition, right, that we might need in order to account for the suppression of the afferent input. And I'll note that if people know the work that was done by people like Curtis Bell and more recently Nate Sawtelle in electric fish, this is different than what people see in the electric fish model, where if you paralyze the animal and you look at just an efferent's copy signal, you can actually get this negative image. So the mechanism that's used in monkeys for reafferent suppression seems to be more sophisticated. We also did an experiment where we looked at higher level input. So we had trained a monkey to drive. We gave him a little steering wheel, and here he's driving with his steering wheel. We gave him a target that he controls that's on top of his turntable. So he turns his wheel, the turntable moves, and his job is to follow a goal target that we control. We move the goal target with a sinusoidal trajectory horizontally back and forth. So he's trying to follow that. He's doing a pretty decent job. And we were sort of disappointed in this condition. It's a higher level task that the neurons actually responded as if it was a passively applied stimuli. And I'm happy to take this up again at the end during the discussion period, because there's some things that we might do in this experiment that would actually give us some insight, because this is actually has a number of features that make it quite a high level task. So our current hypothesis is that we actually only see vestibular reafference suppression or cancellation when the sensory feedback that's experienced by the brain matches that was expected as a result of the motor command that was generated. So that's the case during active normal movement. So you generate a movement based on your experience. The brain has some expectation of the sensory feedback it expects, and if that matches what actually comes back, then we get suppression. And this should start to sound a little bit like motor learning, right? And the idea is that the brain, and in particular the cerebellum, computes an estimate, or what's called an internal model, the sensory consequences of action, which it compares to actual sensory feedback. And the difference between the estimate and actual sensory feedback is called a sensory prediction error. And you use a sensory prediction error to update the motor program. So in the present context, the computation of sensory prediction error is actually what's enabling the distinction between vestibular X afferents and reafferents. So this is work that was mentioned in the introduction done by Jess Brooks. So the idea is we record from here output neurons of the cerebellum, particularly the anterior vermis, in the rostral fastigial nucleus. We can also record from the vestibular nuclei and look and see what happens and are their responses consistent with this idea. So again, our hypothesis was that we would only see suppression when we have this match between expected and actual sensory feedback. So the idea is if this is true, we should not see reafferent suppression when we alter the relationship between the motor command that was generated and sensory reafferents. So to do that, we change the relationship between the motor command and sensory reafferents by applying resistive torque. So we had the animal normally making his normal active head movements, and then we applied those kind of replay passive movements like we did in the past. And then what we did is, after we had our control data, we put on this load, this resistive torque. So when the animal tried to move his head, he only could move his head about half as much initially. But over time, in order to get his reward, he learned he needed to generate a bigger motor command, and then he reattained his normal active movement. And then once he was in steady state and had learned that, occasionally we'd take the load off and have catch trials, and then at the end we could just take the load off completely and look at extinction. So what do we see? We see that these are two columns here represent what I've already shown you, normal passive and normal active movements. Here's the learning. We see that initially the head movement's about half of what it was. Then we have middle learning, and by about 50 trials, we see head movements look a lot like they did before the load was applied. So the load is applied through this whole gray box. And then we take the load off every so often, we can get these catch trials where the animal is now updated its internal model on the basis of this load, and of course has a bigger motor command, so he will overshoot and generate these big head movements. So what do the neurons do? The first two columns you know already. You have big responses to passive, but not such big movements to active, responses to active. So you get this big suppression. And then what's really interesting, right, is these head movements are about half the size of, or less, of the original active movements, and they are active movements, but the neuron responds a lot more. And in fact, it responds with a sensitivity that matches that of what you would expect based on its passive sensitivity. And then halfway through learning, you get about halfway suppression, and then back when, even though the head movements are bigger, right, and these are all active head movements, here the monkeys relearned the load, and he's now updated his internal model, and we see this reemergence of vestibular reafferent suppression. So perhaps an easier way to look at this is in terms of normalized sensitivity relative to the passive sensitivity. So we have passive sensitivity for control movements, and then this great reduction for reafferents normally. And then over the time course of learning, even though the head movements get bigger, the sensitivity of the neuron gets smaller as the reafferent suppression is reemerging. Right, so initially the neurons encode the stimulus as if it's passive, and then we see this graded return to a reafferent suppression. And then again, these catch trials, which are really big active head movements, are encoded as passive movements. And what's really nice is this actually follows, you can think of this in terms of head velocity error compared to the sort of control active movement. And the time course of this reemergence of reafferent suppression follows nicely the reduction in head velocity error. I can also show this on there, rather than breaking it into three phases. We can do this trial by trial, and we get, again, this nice relationship between the reemergence of reafferent suppression and head velocity error. So what I just showed you there is when the relationship between the motor commands to move the head and resulting action is altered, neuronal responses are consistent with the updating, and it's very rapid, of an internal model. So this is critical for the sort of argument I made initially with the fact that you want to be able to differentiate between active and passive motion, because sometimes if a motion is unexpected, you're going to want to mount this very robust vestibular spinal reflex response. Whereas if it's a motion you expect, you would actually not want that. It would be counterproductive. Okay. So I'm just going to finish up. I will skip this because I think I'm running a little late, and just note that we've also done some experiments. So one of the things about the vestibular system, it helps keep track of where you are relative to gravity, and that's a really critical feature of the vestibular system to keep us from falling and keep us upright and give us a sort of anchor relative to the world. So we also looked to see whether neuronal responses were consistent with the internal model that predicted the sensory consequences of gravity. And if people are interested in this, this is a recent current biology paper with my postdoc Isabel Macros and Jerome Carriot again, where we looked at how neurons respond in passive conditions. Net acceleration is what would be sensed by the otoliths, for example, and there are neurons that in the vestibular nuclei that sense net acceleration. There are also some neurons that actually combine otolith and canal input to compute tilts, and others translations. So these tilt and net acceleration neurons are encoding sensory consequences of gravity. And what we find as soon as you actually have the animal generating active tilts where the head is moving relative to gravity, again, we see this suppression. And in contrast, the afferents, again, are really context independent. Their responses remain robust and unchanged from passive conditions. So the idea is that response of these cerebellar output neurons are also consistent with the existence of an internal model that predicts the sensory consequences of gravity. And we're moving now to look at other behaviors with our central neurons and cerebellar neurons like walking, and we're using deep lap cut. And we're collaborating with Tim Harris using a non-human primate version, a beta version currently of a neural pixel probe. It's not as with fewer channels, but to try to develop technology so we can do this type of work in non-human primates and ultimately use these probes for more human translational work. I'm just going to finish by noting that I focused on vestibular spinal pathways and made the point that that you want to make this difference between expected and unexpected motion, and use this cartoon as an example of why, or schematic. But also the really critical point is when a unexpected sensory input becomes expected, right, which is the case in that experiment I did with the load, the brain's internal model is updated to re-enable the distinction between reafferents and exafferents. And that's really important. So the idea is if you have a one-time mismatch, the brain's going to encode that as exafferents. But if that mismatch is persistent, then the brain's going to update its internal model. So you can effectively use that same classic motor learning sort of control diagram, right, in order to understand both the computation of exafferents and then the updating based on that sensory prediction error when you have a persistent mismatch of an internal model. And that's important. It's not just something that happens in motor learning experiments, but it's important for the calibration of motor circuits in our everyday life, right? As we swing a tennis racket back and forth, our muscles are always changing and we always need to be calibrating our circuits. I'll finally note, and it's just take me 1 minute, that this basic principle is also important for perceptual stability, as I mentioned. And I should note that Oman Zuberi, who's a current finishing graduate student in my laboratory, has been actually looking at the computations within the cerebellum proper and recording for Purkinje cells to understand how is this, what kind of insight can we get into how the brain is constructing this internal model during self-motion. And his work is ongoing, and I look forward to presenting that in my next talk. Okay, so perceptual stability is something we've also looked at in trying to understand what information is encoded by ascending the thalamical cortical pathways. That's work done by Alexis Dale, who was a PhD student in my lab at McGill. And for those experiments, they look very similar to what I've shown you already. We recorded, instead of down in the vestibular nuclei or the cerebellum, we are now recording from the VPL, which is the ventral posterior lateral thalamus, which is the main relay of information up to areas in the cortex, as I mentioned, that encode self-motion information. These should look quite familiar to you. These traces on the right, we have the dashed line would be our externally applied rotation of the turntable. The black line is total head and space motion. The red line is the motion that the monkey's generating. So we have reafferents in red, dashed line is exafferents, and then black is total head motion. And what you can see again is the neuron is selectively encoding the afferents and ignoring the information, vestibular information based on the motion of the monkey's self-generated movements. So since these neurons are getting their information from the vestibular nuclei, maybe it's not that surprising that they're selectively encoding unexpected head motion or externally applied head motion. And if we look across our different structures, again, afferents don't make a difference. This shows for both rotations and translations. We get maybe 70% attenuation in vestibular nuclei neurons, also in cerebellum. And then thalamus, it's even tighter. We see almost 90% attenuation. So things get more and more precise with respect to encoding unexpected self-motion. So I'll just finish here with a note that what we showed you is that descending pathways, or what I showed you is descending pathways. It's important to make this distinction between active and passive motion for calibrating motor circuits and behavioral dependent modulation of vestibular spinal reflexes. And then in terms of ascending pathways, perceptual stability is important to keep track of what motion you purposefully made versus what movements are unexpected. I always make the analogy of an earthquake. I've experienced some very minor earthquakes, but they're very salient events. You really know that you did not move and something else happened, and it's a very strong sensation when you feel that, even though the stimulus itself might be very, very minor. So in summary, what I've told you is there are two distinct information channels in vestibular processing, regular and irregular afferents, and that's true whether information is passively applied or self-generated. Neurons at the first central stage of processing encode X afferents, and their responses to reafferents are greatly suppressed. Vestibular sensory responses to dynamic self-motion and the consequences of gravity are canceled when there's a match with the brain's expectation. This idea of cancellation of sensory reafferents. And that the output of the cerebellum reveals a very rapid updating of an internal model that we can see on the order of 40 to 50 head movements as unexpected sensory inputs become expected, and also the distinction between active and passive vestibular stimulation encodes perceptual stability, as we know from our thalamic recordings. And I think maybe I'll stop there. Let me see how I get back into... Can you guys see me now?
Yes, we can see you. Thank you very much for the very, very interesting talk and very interesting experimental paradigms and setups that you have. So we are almost out of time for the main talk, but maybe we can have only one question in the main session, in the main hall, if there's any question. And if not, I would suggest that we just move to the panel with Dr. Cullen. So with the people who signed up for the panel, then we can continue the discussion on the results. Is there any question? If there's any question, please feel free to raise your hand or type it in the chat box.
Yeah, it's a very simple question for general context. I mean, there is a puzzle I would have added at the very end, which is that I started by saying you need the vestibular system to generate a VOR as you move through the world, right? And then at the end, I show you that the vestibular system is actually suppressed during active movement. So the question is, how do we solve that? And I just note that the vestibulo-ocular reflex pathway doesn't show the same feature. So it's not a trivial feature of all vestibular pathways, right? This is very specific for vestibular spinal and vestibular thalamic, but the vestibulo-ocular reflex pathway you'd actually want to be active as you move through the world.
That was actually my question. So because part of the function of the VOR is that when I actively move my head to be able to stabilize my eyes, and so this is a separate nuclear that you're recording from that can take. So in the VOR system, you would expect both passive and active, or mainly active, maybe part of the motion to be canceled, right? Maybe passive stays, or no?
Yes, it's the same nucleus, but you have different classes of neurons within that nucleus that you can actually make out based on constellations of properties, and people have actually done intracellular staining and traced them. So the VOR neurons are a specific subclass, which we've also studied, and they're actually, as soon as if you want to move through the world or do anything where you want to keep your gaze stable, they're doing their job. But if you actually want to redirect your gaze in space, say you move your head to move from one side to the other, head and eyes, you actually turn them off with a gaze command. So if you want to redirect your gaze, that's when they're counterproductive. So just like you turn off vestibular spinal reflexes when you want to move through the world, you turn off VOR neurons when you want to redirect your gaze. So it's, from an engineering point of view, totally logical.
Very good.